Today we will be discussing epistasis and how this relates to the first prompt for the second genetics mini video. So what is epistasis, you might ask? Epistasis is the expression of alleles at one locus that masks or modifies the expression of other alleles at another locus. This results in multiple genotypes that lead to the same phenotype. In order to better explain epistasis, we are going to be looking at trolls today. We will be specifically looking at duplicate dominant epistasis. Duplicate dominant epistasis means that at least one dominant allele at either locus results in the same phenotype. The different phenotype occurs when the organism is homozygous recessive. In this troll example, we see that the skinny phenotype occurs when the individual is homozygous recessive at both the A allele and at the B allele. The diagram then branches off, and if a dominant allele is present at either the A locus or the B locus, this results in a pudgy troll. To exemplify the idea of duplicate dominant epistasis, we crossed an individual that was big A, big B with one that was also big A, big B little b. After completing this cross, you can see that the 9 individuals had big A blank, big B blank. 3 individuals had big A blank, little b. 3 individuals had little a, big B blank, and 1 individual was homozygous recessive, being little a, little b. When you combine all of the combinations with at least one dominant allele, you get a 15:1 ratio, which is the standard ratio for a duplicate dominant epistasis cross. On this slide, a cross of big A, little b parent and a little a, big B parent is shown. This cross is not a dihybrid cross since it is not big A, big B, and big A, big B, which means that the typical genotypic ratio of 9:3 to 3:1 will not work for this cross, and with that being said, the duplicate dominant epistasis ratio does not work either. Here are the references that were used for this presentation.